Okay, so that explains sort of the initiation of the action potential. Something has to depolarize a cell, right? Depolarization leads to firing of the cell, leads to a current being sent, right? The sort of key signature, important moment is depolarization, right? To minus 55. If you get to the threshold potential for that voltage-gated sodium channel, they're going to all open, right? For a millisecond, you're going to have this flood of sodium coming in, and that'll initiate, we call the action potential, the current that's going to flow from one end of a neuron to another. But how does it get from one end of the neuron to the other? Once it comes in, what causes it to continue down the axon, until it reaches the axon terminal? Here's our neuron. This is this process by which, the sodium that enters, continues down to the end of the axon, is referred to as propagation of the action potential. The action potential sort of propagates down, from one end of the cell to the other. I will point out that, in a typical neuron, we have our dendrites sticking out here. We have our soma with the genetic material, and then we have our axon and our axon terminal. But we also have this bulge generally at the beginning of the axon that's particularly important. It's called the axon hillock. It's like a little bulge at the start of the axon, and it is coated, packed with voltage-gated sodium channels and also voltage-gated potassium channels. So the issue is you have to depolarize, right, this neuron to threshold at this axon hillock in order to really initiate an action potential that'll propagate all the way down to the end of the axon. If you have something, a little depolarization occurring way out here and one occurring way out here, you might not get enough of a sodium influx to reach, this hillock and depolarize, right, the membrane here to minus 55. But if you can do it here, if you get a lot of inputs, a lot of firing, a lot of depolarization, if it gets to minus 55 at the axon hillock, you're going to get an action potential propagate down the nerve. So let's look at a little segment of membrane here, a little post hillock here on our axon, and discuss propagation. So here's our membrane, right? Here's our phospholipid bilayer, largely impermeable. Here is a sodium channel. It's a voltage-gated sodium channel at minus 55. So this is, we've reached threshold, and it's in its second conformational state, right? It's open, and there's a channel that allows for sodium to enter, right? And sodium has entered, right? There's a lot of sodium all of a sudden here in this little segment of the membrane, right? And we've reached a voltage, difference or a potential difference of plus 20 on the inside because of the entry of all of this positive sodium. Now let's look a little bit further down the neuron here. If you go just a little bit further towards the terminal, you'll find that you have mostly potassium on the inside, right? And you have sodium really masked on the outside again. And you have that same relationship, that you typically have in a neuron at rest, right? It's minus 65, right? And the voltage-gated sodium channels that you're going to encounter are going to be in that first conformation. They're going to be at the resting potential conformation. They're going to be closed, right? So I want you to think about which way will these sodium ions, this current of sodium, right, move? And then also I want you to think about why will it move? And I'll go over it. So if you look here, you've got a positively charged area, right, inside the cell. You've got a high concentration of sodium. A little bit further this way, you have a negatively charged internal, space. And you've also got higher concentration of potassium, not very much sodium at all. So which two forces will drive sodium in this direction? Hopefully you've said electrical force and concentration gradient force. And so the sodium will move. It'll be attracted in this direction, right? And as it comes in here, think, it'll actually, be carrying in positive charge, right? So it's minus 65 here, but as this sodium current moves in, what does it do? It depolarizes it, right? It'll depolarize it to minus 55, and at minus 55, this next crop of, voltage-gated sodium channels are going to open, right? And in will flow sodium to help keep that current, basically suitably staffed with sodium ions. So think about this page and see if that makes sense in terms of action potential propagation.